Okay, right. We're on the last day of our experiment on sea urchin development, and I've just made a wet mount of the embryos from the control group, the ones that have been raised in clean seawater. We looked at these yesterday at about this time, and you should have seen lots of gastrulas. But let's see what happens after 24 hours. I haven't looked at this yet. I'm not entirely sure what we'll find, but let us peruse. Okay, what I'm immediately seeing is a lot of little ciliates. That is perhaps slightly concerning. This is not an embryo. This is just something I'm curious about. Okay, that looks like we've caught a little ciliate in the act of cell division, which is not what I was hoping for, but I'll take what I can get. There's certainly plenty of marine ciliates as well as freshwater ones. And yeah, so there's more. Okay, that looks like that might conceivably have been one, but it's dead and messed up. Okay, that's another little ciliate there. Okay, on this slide, I'm not seeing embryos at all. And it's possible the number of ciliates and those tiny little dots are probably bacteria. The number of those means that we could have lost our embryos to bacterial infection, which does sometimes happen. I'll leave this up for your entertainment and quickly make another slide and see if I can spot some this time. And if that doesn't work, we'll just go to the experimental slide and see what we can see. So swiftly, I'll make another slide. Hmm. Hang on. All right, let's look at this one. All right, good morning. We're looking at our embryos, or hopefully we will, for the second day. And this is also our control group. And again, I'm seeing a lot of little ciliate zipping around. Okay, what I think has happened is that we got bacterial overgrowth, and that seems to have offed um, the embryos here. I could make more slides, and I might do that once we're done. I might do that on my own time, but at the moment, I'm not really seeing anything. So it looks like none of our embryos made it. Um, but there's a lot of bacteria and a lot of little things feeding on those bacteria. So I don't think it's because our embryos had developmental problems. I think that we just got infected. I will take a look at the other one, the experimental group, and we'll see what we got. And even if we don't have anything in the control, you've still got pictures of what Pluteus larvae should look like. And if we get anything in the experimental group, you can still compare what we see to those. So give me a moment to make a slide of the experimental group with the nickel. Yeah. Add the Vaseline. All right, looking at the experimental group now, this is the one that got zapped with nickel. So let's see if we have anything in here. All right, we've actually got a few. So let's see what we can identify. That's interesting. Okay, that is probably an egg, or it could be an embryo. Either way, all of the wriggling stuff inside, that's probably little particles getting bounced around by Brownian motion. There's also a little ciliate feeding on it. So actually, some of those could be bacteria too. Yeah, I think that's an egg that's being attacked by bacteria, and we probably shouldn't count it. It's pretty pathological, although it does look pretty weird. Um, okay, I think that's another embryo that has gotten messed up, but again, it's being swarmed by, yeah, again, I think that's bacteria that are macking on it, as it were. Okay, this one's still viable. You can see the gut if we focus up and down. You can also see that it's not round anymore. It's got a definite triangle shape to it. This is not a pluteus yet. It's what's called a prism larva. So what's happened is that the gut has developed. I think the mouth end may be what's currently up at it's about your 2 o'clock right now, and you can see the gut kind of extending down below it. Cilia are enabling it to swim, and the shape is starting to transform from circular to kind of a pyramid shape. So this would count as a prism larva, Prism. So we've got one. Okay, that's another one that's gotten hit with bacteria. And another one. And there's another one that I think is too messed up. And okay, there's okay, that's another one that's also got bacteria macking on it. Well, there's one to the left that I think is too wrecked to tell. But in this one, you can actually see the complete gut. There's kind of a central stomach, and then as you focus up and down, you can see the mouth ends at about the 11 o'clock and the anal end at about 5. And again, the shape is no longer really round. This would also be a prism larva. However, it does have something sticking out in front of it. And I'm not sure what that is. That's not a normal prism larva. Um, no, I'm not sure whether that's because of the nickel or because it's gotten bacterized. We have the problem that the bacteria are going to mess with things as well. But we've got at least 2 prism larvae, and one of them is doing something weird, growing that schnoz sticking out on your 11 o'clock. That is not the way they normally look. Unless it's just trying to burst out of the fertilization membrane, in which case it might not be as big a deal. Okay, hey, I might have to take that back. It may actually be trying to break out of the vitelline or fertilization membrane that's been surrounding it all this time, because I think I saw it make a muscle contraction just a minute ago. Let's see if it does it again. Yeah, I think it's doing it. I'm not touching the focus knob right now. So any movement that you see is the embryo doing it itself. There we go. Okay. So it's developed at this point enough to have functioning muscle cells or contractile cells anyway. And I can see a complete gut. You've got that central stomach right there that's in focus now. But there's little sections of it in front and behind. Like right now, I can see the 5 o'clock. There's a second little circle right there, and there's another one at about the 11 o'clock. And I think those are the anus and the mouth, respectively. So we have got a complete gut now. This thing has completed gastrulation. And yeah, I think the reason it's got that funky shape may be that it's trying to get out of its membrane. Which means that we can probably get away with calling this a prism larva, even if I can't quite see the full shape. So there. So we got that much. Okay, tell you what, I'm going to make one more slide from the nickel, see if we can garner any more data. I'll leave that up for the moment, but I'm going to do one more slide. All right, hang on, Jamaica. I see you. All right, I'm going to look at one more slide. Okay, good morning, Jamaica. We're looking at the sea urchin embryos after nickel exposure. Okay, and this one has quite a bit more. All right, I'm going to start panning around, and I think this has enough that we could actually get some numbers on this. So let me see here. Okay, that's a prism larva, but that one's pretty messed up. Um, Although, again, that could be all the bacteria zipping around it. I'm going to skip over some that just look too abnormal to even be worth counting. Because, again, it's going to be hard to tell whether an abnormal larva is due to the nickel or due to the bacteria. That one looks to me like a gastrula. I think I can see that central gut. There's a bunch of others in this field of view that just look like junk. Yeah, that one I'm not sure what to do with. Okay, that's a prism larva. And you can see the complete gut. It's kind of extending from roughly 4 o'clock to 10 o'clock. Again, there's bacteria all over it. It doesn't seem to be doing very well, but that is a prism larva. All right, I'm seeing several more. You may not be able to see them all because my field of view is wider than yours. But I'm seeing more that just look screwy, although, again, I can't tell the difference between screwy because they're bacterized, and screwy because they're developmentally screwy. But there you've got that's another gastrula. It's interesting. That's not terribly different from the ones I was filming yesterday. So at the very least, development doesn't seem to have gone on much farther in this one than it did, I guess, yesterday afternoon. There's another one that just looks like a mess. There's two more that are messes. Okay, when we do this write up, we're going to leave out the ones that are messes because we can't tell what the factor is that's causing that. So there we've got there's a prism larva. And again, I guess that schnoz right there maybe is part of normal development. You can see several portions of the gut as we focus up and down. So we can count that as a prism. We can count that as a gastrula. Yeah, we can count that guy as a gastrula. I don't know what we can count this one as. He's not covered in bacteria. He's just kind of sitting there lumpy. So, yeah, I'd call this one just generally abnormal because I can't see a gut. Yeah, ditto for that one. Yeah, there's a whole lot here that look abnormal. On the face of it, I'm tempted to blame that on the nickel, but it's just hard to say whether it's the nickel or bacteria, which would go with things like, low oxygen levels in the water. It's just hard to say what's causing that. So that's a gastrula. I think we already saw that prism larva. Yeah, there's a bunch of weird things right there, but there you can see the bacteria just chowing down on it. Okay, I think here we might be getting a prism. Either that or a late gastrula. I'm not sure that the gut's gone through, but you can see that dent at roughly the 1 o'clock position and then extending down. So I might be tempted to call that late gastrula. Okay, there's another prism larva with that schnoz sticking out, where the contractile cells are in fact starting to contract. And that might be the mouth up there at the front at about your 8 o'clock, with the gut going completely through. So yeah, I'd be willing to call that one prism. And that one's just covered in bacteria and schmood. And that's yet another prism larva. And you can see the gut there. I think that'd be the anus at about your 10 o'clock. And then pointing down in the 5 o'clock direction, that's your gut. I think it is. And of course, I'm right because I have a PhD and you don't, so neener, neener. Okay, that one's pretty bacteria heavy and it just looks kind of like a mess. And this thing is definitely a hot mess, but again, it's so covered in bacteria that I don't know why it's a mess, but it's definitely a mess. There's some others I'm just passing through just because they look so awful. That one looks like it made it to the prism stage, although again, the bacteria are chowing down. That's one that just looks horrible. Maybe prism at a funky angle. And if you call that prism, I wouldn't flunk you this time. There's another one that just looks wrecked. And another one that could be prism, but the insides don't have a clear gut. The insides just look messy. And another one that I think is just garbage. And okay, I'm going to call that one a late gastrula. It doesn't look too damaged. This one's actually still alive enough that it can swim on its own. Yeah, so there you can see the blastopore. And okay, I'm seeing some, it's kind of turning. So there's that schnoz that it's got. Whoops, come on, Wagoner. Come on, stay with it. Okay, yeah, that's actually a fairly healthy prism larva. As it turns, you might be able to get a better idea of the shape. And I'm sorry, I'm trying to keep it centered. And it's not easy because what I'm seeing on the screen doesn't perfectly match what's on the what I'm seeing in the eyepiece. And trying to keep it centered on the screen while it's moving in two different directions, depending on where I'm looking at it, is kind of mentally weird. But there, okay. All right, that'll have to do. But I think you saw the complete gut. And there it is again. That's the same guy. Ah. Yeah, there's the And now he's kind of turned around. Yeah, but that's the anal end. All right, it's a prism larva. I'm going to quit chasing it. And there's another gastrula. Yeah, if it's still primarily round, I'm going to call it a gastrula. If it's starting to show that conical or pyramid shape, we can call it prism. And that got to gastrula stage before the bacteria zapped it, I think. And that's the right shape to be a prism larva, but I can't see a clear gut on the inside. So if I had to call it, I'd guess something went wrong with gastrulation there. Okay, that's another prism larva, and that one not moving. You can see the anal end at about your 11 o'clock. Maybe I did show you that one before, but that one you can see that the gut's complete. If I did show you that one before, don't count it. So we're almost done. Okay, that's another prism at kind of a funky angle. That's a prism. Okay, and there's a couple here that just look like a mess. That's a prism that something has gone kind of wrong with. That looks like a rather screwed up gastrula, is my guess. That one's just plain screwed up. As is that one. As is that one, although that one is just being swarmed. Okay. There, that one seems to have a gut going through, so that would be early prism stage, I think. There's another prism. And that one, yeah, you can see the anus down at your 6 o'clock, and the mouth is coming in at about the 11 o'clock. So there's that complete gut. And the whole larva is, taking that kind of pyramid shape. And from this, if it were going to transform into a pluteus, it would grow some arms. But that has not happened yet. And that I'm going to call still a gastrula, because the shape is still circular, and I can't see multiple divisions of the gut. And I've got, this one here just looks trashed. And that one just looks trashed. And that one looks trash. That looks like a gastrula that couldn't figure out what was supposed to come next. Okay, and we might have seen that one. We might have seen that one already. All right. Okay, what I'm seeing is that we've got in the nickel tank, we ended up with a lot of screwed up larvae. The problem with interpreting that is, first of all, because the control tank got so full of bacteria, I don't really have a normal to compare it with. And second, there's enough bacteria even in the nickel tank that it's hard to say how many of these screwed up larvae might be, screwed up because they're being eaten by bacteria, and how many of them might be screwed up because of the nickel. We've got two variables here that I can't really disentangle. What I do have, which I should have included in the handout, but I will send it out to you separately, is we do have data on how fast development is supposed to go for larvae being kept at room temperature. And at the very least, we can use that as a control. We can compare what we're seeing in our nickel larvae to the tables of normal development times and see if the nickel larvae are developing on schedule or if they're developing too slowly. You can also at least record the number of larvae that we saw that look that look weird. There was a bunch that I said were trashed or otherwise abnormal. Although again, it can be hard to tell if they're degenerating just because of bacterial attack or if they're degenerating because of the nickel. But at least we can present that, even if it's not data as nice as we would like it. So what I'm going to do is stop the recording for now and declare this experiment a partial success.